Good afternoon, everybody. Thank you for joining us to this exciting webinar. I'm Elisa Bandera. I'm a professor of epidemiology at Rutgers Cancer Institute of New Jersey and currently chair of the Lifestyle Behaviors, Energy Balance, and Chemoprevention Special Interest Group. And in the call is Marji McCullough. You can see, wave, Marji.
Hello.
She's the vice chair of the SIG currently, and she's a senior scientific director at the American Cancer Society. We have in the screen right now the meeting, the next meeting for ASPO. Please mark your calendars. We have a very exciting program, March 9 to 12 in Tampa, Florida, and hope to see you all there. Cheryl, if I can show the webinar, or you can show you if you want, the lineup for the, Cheryl?
Yeah, I said I didn't have that slide.
Okay, so you let me share, I can share it so that we can. So this webinar came actually from the, as a feedback from the survey that we did in March, and a lot of ASPO members were very interested in metabolomics. So we are very excited to have this program today. We have a wonderful lineup of speakers, and I'm indebted to the three of you, but particularly to Dr. Scalbert and Dr. Ferrari, because they're actually calling from Lyon, France. So it's 9 p.m. for them, so we really appreciated their willingness to be there for us. Cheryl, okay, I will introduce it. Hopefully we can, Cheryl, if you let me share the screen, okay. So I can share my screen, hopefully you can see it. Can you see it, the screen with the lineup? So we have 3 speakers, and each one of them is going to talk for 15 minutes. After them, there's going to be a Q&A question that Marji is going to moderate. The way it's going to work, if you look at your Zoom program, in the bottom, there's a Q&A button that you can click, and then you can type out the questions. Anytime during the webinar, you can do that. And at the end of the program, Marji will read the questions. We'll try to get through as many as we can. We want to stop at 4 p.m., so hopefully we will get most of the questions answered. And one of our experts will answer the questions for you. I would like to introduce the 3 speakers now so that the webinar flows better and goes from one to another without having to come back to me. The first one is Dr. Augustin Scalbert. He's the head of Biomarkers group at the International Agency for Research on Cancer. And he's going to be talking about metabolomics in nutrition and cancer epidemiology. Next, Dr. Pietro Ferrari is going to talk about analytical strategies and challenges, and he's also at IARC. He's the head of nutritional methodology and biostatistics. And the third one is Dr. Steve Moore. He's an investigator at the National Cancer Institute, and he's going to be talking about recent cancer findings. And after the 3 speakers, again, we will have the Q&A session. So without further ado, I would like to pass the screen to Dr. Scalbert. Thank you very much. Oh, I have to stop share, okay.
Okay, so hello, I'm Augustin Scalbert from IARC in Lyon in France, and I'm very pleased to contribute to this webinar and to share with you some elements about metabolomics and application of metabolomics in cancer epidemiology. We've been busy at IARC for the last few years in developing some capacity to apply metabolomics to large cohort studies. So I don't know very well who is in the audience, but I will start with one slide to introduce metabolomics and explain briefly how it works for those who may not be that familiar. So on this slide here, the idea of metabolomics as actually for various omics technology is to collect a large amount of information, and thinking about a lot of information, then you can easily understand by comparison with metabolomic fingerprints or metabolomic signature, that if you put enough information in the data you collect, then it will be easier to characterize precisely a phenotype and to make a difference between one subject and another one. Now, when we apply metabolomics in some of our studies, we do not work on obviously one or two subjects, but we work on a group of subjects like A and B, and we compare this group of subjects. So we first start with study design. We collect some analytical data, and this analytical data can be collected with various techniques. I will give some details on that. Then the data are pre-processed and analyzed with statistical methods that will be detailed by Pietro Ferrari. Then the data are interpreted, and at the end, what you aim at is to identify new biomarkers. They can be, for example, early biomarker for disease. They can be biomarkers of exposure to a risk factor for cancer. And also with this big amount of information you collect, you can also learn about biological mechanism, looking at the metabolites that are under or overexpressed, and learn and understand what is finally, or trying at least to understand what leads to cancer. Now, metabolomics is just one dimension in the different omics. So you see on the left the genome, transcriptome, proteome, and metabolome. And so the value of the metabolome is that it is the most downstream dimension of metabolism. And being that, you can understand that it may capture a lot of these environmental influences that are very important to understand the etiology of cancer. Now about data acquisition. So initially, metabolomics really started with nuclear magnetic resonance spectroscopy. And there has been a lot of work published with this technique, and still many people are using it today. One limitation of NMR, as we say, is the limited sensitivity. So in total, you see that it's mainly 20 unique metabolites that can be measured. This can be various metabolites, including amino acids, organic acids, amines, lipids, lipoproteins. And more recently, the mass spectrometry really developed very quickly in the field of metabolomics. And one of the main reasons is the very high, or the relatively very high, sensitivity of this technique. And we're not talking about a few metabolites, but clearly about hundreds or thousands of metabolites. And on this graph on the right side, you see in green, and this is development and the number of publications that has appeared over the last few years with mass spectrometry, using mass spectrometry. Now there are two different approaches when we think about application of mass spectrometry in the metabolomic field. We initially started analysis with classes of compounds, and there are specific methods that have been developed to analyze, for example, fatty acids or acylcarnitine or bile acids. And you see the number of compounds that have been measured in this method. And with metabolomics and targeted metabolomics, some metabolites are also selected a priori, and the method is developed to quantify these different metabolites. So I just mentioned here some references of methods that have been published and that give a good idea of what is possible to do today with targeted metabolomics. We use in the laboratory a kit that has been developed by a company in Austria that allows to analyze these different classes of metabolites. Now, targeted metabolomics is limited by the number of metabolites. Here on this table, we speak about 150 to 250 metabolites, and it's limited in the number of metabolites because for each we need chemical standards to draw some calibration curve in order to quantify these metabolites. Now there is another approach that is called untargeted metabolomics, and with untargeted metabolomics we can aim at much broader coverage of the metabolome. So this is how it works. You use, and this time, some high resolution mass spectrometers, And you can apply different techniques and to maximize the number of metabolites. You can use your mass spectrometer with positive or negative ionization. You can use different types of chromatography. And then in a given batch, you can analyze up to 600 samples, and each sample is characterized by a chromatogram that you see here. You differentiate two different groups, like cases in red or controls in black, and you apply your statistical method to identify the metabolites that make this profile different. And then once you have candidate biomarker, these candidates are characterized by accurate mass and retention time, and you can screen for this accurate mass in a chemical database. This database can be also developed in the laboratory, and you have here an example of a database with 300 chemical standards, and then you can compare the behavior in your method of this standard with what is detected in your chromatograms. There are different approaches also with untargeted metabolomics. You can run your statistical analysis only on the annotated metabolites that have been confirmed with your in-house database. At the maximum, usually you see 800 metabolites, usually not more. Now, we can detect more metabolites, but many of these metabolites are unknown. I mentioned this figure, 6,000 mass spectrometry features. And then once you have identified some markers of interest, you can screen a metabolite database to recognize the metabolites that you have detected. You can also use this data set to analyze only a few metabolites of interest. Let's say if you are interested by meat intake, you have metabolites for meat intake, and you can selectively extract this information and study association with the risk of disease. In this table, you have a summary of the different prospective studies on cancer that have used different metabolomics techniques that have been published so far. So these techniques have used either NMR or mass spectrometry. And you see that some have used the service of a company that is called Metabolomics. This is a very well advanced company with 800 metabolites characterized in untargeted metabolomics, or have used the Biocrates kit I was mentioning earlier. You can use also the technique with untargeted metabolomics, as we did in several of our studies, like in hepatocellular carcinoma. And I will show you some results here that we obtained recently. So in this particular experiment, it is run in the large European prospective investigation on cancer and nutrition study, a large European cohort with 500,000 subjects. And in this study, we selected the cases of hepatocellular carcinoma and compared them to match control. We analyzed the corresponding plasma samples by high-resolution mass spectrometry. This was the information that was collected. And then we found 92 discriminant metabolites, and some of them could be annotated, 46 that could be annotated. And you see some the top hits and that are shown in this table. It includes retinol, vitamin that has been related to cell proliferation in particular, and that was also observed in and also reported in a previous study in the ATBC cohort. You have some steroid hormones, you have some lipids. This is well-known metabolites of lipid in liver, metabolites of vitamin E. You have some amino acids, some bile acids, and a possible indicator of DNA degradation. So with all this information, then we can make some hypotheses and test these hypotheses in other models. Now I will talk about biomarkers of exposure. We were very interested, and we are very interested by the exposome, or what we call the food metabolome. The food metabolome is a sum of all the metabolites that are derived from the digestion of different foods. And so each food brings some characteristic compounds that are digested and absorbed through the gut, and finally found in the circulation and analyzed in blood or in urine samples. We are interested to characterize the food metabolome and to identify among all those compounds that are found here, those that can be useful as dietary biomarkers. So I will give you two examples. One example about food of plant origin. So you see a selection of foods like coffee, red wine, citrus, fruit, tea, and so on. And so we applied this technique to some urine samples that were derived from the EPIC cross-sectional study. They were analyzed by the technique that I've described before, and we found in total 2,000 signals that were associated with the intake of any of these 6 different foods. We were able to identify in total about 40 different markers associated with the intake of these foods. Could run some RUC analysis to find the best predictors of food intake, and you see these different examples on the figure on the right side here. Another example about markers for meat and fish intake. This time we used samples that were derived from a controlled dietary intervention study where subjects consumed different types of meat or of fish. And we identify a number of metabolites that were, for example, for those indicated in red here, there are some metabolites that are characteristic of the intake of chicken. And we found some metabolites that are characteristic of fish intake, and this is trimethylamine oxide that have been very much discussed in literature and often reported as marker of meat intake, but our results show clearly that it's a marker mainly of fish intake. We found some other markers that are more generic markers for the consumption of meat or fish intake. Now, all this information is developing quickly, and many labs are now using this metabolomic approach to identify some markers of dietary exposure. And so we collected this information in a new database that is called Exposome Explorer. This database is freely accessible online. And we already have in this database about 500 different biomarkers with a lot of information of concentration, cohorts, or population where these markers have been measured. Some important information like reproducibility over time when you have a unique sample to measure a biomarker in a large population. The measurement should be representative of the individual. Now, these markers, once identified, we are interested to study their association with the risk of cancer for ourselves in some prospective studies. And I just give you some references here, some work that are used, a metabolomic data set, to mine this data set and identify and study the association of different markers of exposure with the risk of cancer. So you see, for example, here, this is not a metabolite derived from food, but from smoking, positively associated with colorectal cancer risk. No association with this methylhistidine metabolite, the marker for chicken. Trigonelline is a well-known now marker for coffee intake, inversely related to colorectal cancer risk. On this slide here, I just want to illustrate the issue of the number of samples that can be that has been analyzed in various studies with this different metabolomic approach. So to plot this graph, I selected publication that I've used the analysis with different commercial assay or commercial platform. So I mentioned this kit with the Biocrates kit, so up to 7,000 samples analyzed in a particular study with NMR spectroscopy and this company, BrainShake, about 25,000 metabolites. This company has project to analyze now 500,000 samples. I would say, and I just mentioned these first two examples here, because when the metabolites are quantified, there is a priori no limit in the number of samples that you can compare. It's not the case for untargeted metabolomic, and many people are working in the lab, some people developing metabolomic, to maximize the number of samples that can be compared using untargeted metabolomics. The reason for that is that the data that you collect with untargeted metabolomics are semi-quantitative data. They are relative concentration, and so you can compare samples within your batch. You cannot compare easily these samples to samples that have been measured in another batch or in another study. These are my few conclusions. So first, metabolomics is a promising approach for discovering new risk factor and mechanism leading to cancer. We still have relatively limited number of prospective studies that have used metabolomics so far, but we expect this number to grow very quickly today. The techniques are not yet fully mature. We still need to standardize these techniques to be able to compare results from different cohorts, and major progress are still needed to improve our capacity to annotate the signals of interest. And lastly, this was my previous slide, the number of samples that needs to be further increased for untargeted metabolomics. And so this is my last slide, and I would like to introduce the nice group. We have at IARC to run metabolomic analysis. All these colleagues that you have on the picture combine quite various skills from analytical chemistry to bioinformatics or epidemiology. We also collaborate with all our colleagues at IARC who are epidemiologists or nutritional epidemiologists. We collaborate with obviously Pietro Ferrari, who will speak in a minute, for biostatistics. And we collaborate with many epidemiologists from the EPIC cohort and from many other countries here. And with that, I would like to thank you for your attention. I'll be happy to answer any question you might have.
Okay. Elisa, can you confirm you see my title page?
Yeah, Pietro, I can see it, yeah.
Okay. So, hello everyone. The moon is shining here, and I'm very happy to talk to you now. So I will talk about the context of the omics data and specifically on metabolomics. I will try to persuade you that sometimes this is a very overwhelming information. I will tackle a couple of analytical strategies for cancer research with some application from the EPIC study that Augustin alluded to. So this metabolomics is a comprehensive and quantitative analysis of a large number of metabolites from biological samples. And the good news for cancer epidemiology is that this technology is progressively available in large-scale epidemiological investigations. And clearly is an incredibly potentially very useful tool for the identification of causal pathway for cancer development and clearly other chronic conditions. So I think there is need for statistical methodology to optimize the use of such a powerful tools. So how do we handle omics data? Well, I always say that before doing anything else, just spend some time to explore what drives variability in large dimension data. So take a look at this plot. This is a heat map. It's basically is a correlation matrix of Biocrates kit metabolites. So this is 150 variables here and here. On the main diagonal, you have the correlation coefficient, which is equal to 1, and then when you have the bluer the squares, the larger the correlation coefficient. And I think from a very simple plot, there is a lot to learn. This is Biocrates kit, so this is a targeted metabolomics, and so you can see that these data have a hierarchical structure. So you can see that the metabolites are subdivided in families, and then you can appreciate that very, very neatly. So a second question is, what drives systematic variation in omics? I mean, can we distinguish random variation from systematic variation? And if there are sources of systematic variation that we don't want to deal with, we need to be ready to A, identify it, B, possibly remove it. When I'm talking about potential sources of variation, I'm thinking about general characteristics of study participants, so age, BMI, smoking status, for example, but also variables that affect our samples when we treat them. So I'm thinking about variables like serum clock contact time, the length of storage, the fasting status. So this is a very common and very utilized way. People use principal component analysis, and they display on this graph, for example, two components in terms of one specific study subject characteristics. In this case, subject gender, and this is absolutely doing a good job. The problem with this plot is that we can appreciate only one characteristic, gender, and we can plot only two components at once. So we have developed a tool to analyze variation in large dimension data. So how does it work? Well, in short, one run principal component analysis, then retain a number of components equal to Q, so which is a lot lower than the original number of variables. Once you have the components, you can regress them on a set K of explanatory variables, and these could be either sample characteristics or laboratory variables. And for each explanatory variables, we determine the partial R-square statistics, which basically summarizes the amount of variability that each factor contributes to explain to the ensemble of the data. So this is an example from the EPIC study. Is a pooled analysis where we use controls from nested case control studies. So we pull together data from 4 case control studies, and these are again Biomarkers kit data. So the PCPR2 produces graph plots of this type. So basically, these are all the variables that we were considering. The total R-square is the total amount of variability that these sets of covariates managed to explain. And you can see there are some variables contributed very little, like age or smoking status or fasting. But there are variables that are incredibly relevant, and we need to think about how to treat them. And I'm talking about study. Remember, this is a pooling project of 4 different studies. The variable country, because EPIC is a multi-centric study from different European countries, and batch, which is a little bit of a gentle nightmare of any pooling initiative when we're dealing with laboratory data. So we need to think what to do here. But the PCPR2 is a neat technique to identify what are the sources of variability. So we have carried out a systematic review of the methods that normalize data. We did that with respect to epigenetics data. We were evaluating some methods like the SVA combat, including the residuals. And this was, the evidence was summarized in the paper that you can see here. Now, it turned out that in fact the residual methods it does a very good job. A lot of evidence that is summarized in this paper could be used for metabolomics data. So I'm going to talk now about a second application. Is together with Augustin's group, we were involved in the in a study for the biomarker discovery for alcohol intake. You remember that alcohol is a group 1 carcinogens as classified by the IARC monograph program. And so we had the privilege of set up a two-stage design. We have a discovery phase. We had data from a cross-sectional sample with 451 samples, and then a replication phase. And we were using the controls of the study that Augustin mentioned. So this time around is untargeted features are set by LCMS in both positive and negative modes. So we're talking here about thousands of features to start with. So this is a very neat Manhattan plot, which is very popular in GWAS studies, where we were basically comparing the intensity of each features with alcohol intake estimated through dietary questionnaires. So you could see that the ones in red are the features that pop up to be statistically significant after FDR correction. So 114. So what we did with 114, we were looking at them in the replication phase, and again we did a heat map because we thought, okay, let's take a look at how do these feature look like, and we could see that there are a lot of information that it is around the center. This is a variance covariance metric, so it's slightly different with respect to the one I showed before. But also you can see off the diagonal, you can see there are areas that are very relevant. So the thing is that what we thought is, we were able to match this feature in the replication phase, and then we were thinking, okay, let's redo this analysis. Let's see what is the association with the alcohol estimated through questionnaires. But rather than using a univariate approach, we thought that it would have been a lot more informative to use techniques that could take into account the multivariate nature of the data. So we used a lasso analysis, and then 19 features were identified. And when we were looking at the performance, these entity, with this linear combination of 19 features, had a correlation coefficient with alcohol intake equal to 0.54, and then had an area under the curve close to 0.8. So this is the last application that I'm going to talk. It all started a few years back, while when we were trying to provide develop an analytical framework for the meeting in the middle. So we had on one hand lifestyle exposure and the relationship with cancer, and we were looking at, we thought that the metabolomics could really bridge the relationship between lifestyle and cancer. So what we did is that using the same nested quiz control study on hepatocellular carcinoma, we had the 132 blood metabolites acquired with the Biomarkers kit, and then our lifestyle exposure was the healthy lifestyle index, which is a very popular score, which is made of different components related to the lifestyle. So there is a component made of several dietary factors, then the BMI, the physical activity, the lifetime alcohol intake, the smoking status. Also, we modified that, including hepatitis and diabetes status, because these are known risk factors for hepatocellular carcinoma. So what we were doing, it was we used a PLS method to identify components of the metabolomics that were maximizing the covariance with the components of the healthy lifestyle index. So this was what the components out of the different lifestyle components, and you could see that basically we managed to come out. We were quite lucky, I must say, because this component was negatively associated to BMI, alcohol, and smoking status, and diabetes and infection, and positively associated to physical activity. So meaning that this was really reflecting a healthy lifestyle. And then on the side of the metabolites, so we could characterize this component. So it was negatively associated to glutamate and exose, and positively associated to a number of sphingomyelins, and negatively again associated to the liver function. So when we then try to relate these two components independently in two separate models to the risk of hepatocellular carcinoma, you could see that the healthy lifestyle index was inversely associated to the risk of hepatocellular carcinoma. And the same thing happened to the metabolic signature. The metabolic signature turned out to be very, very strongly inversely associated to the risk of hepatocellular carcinoma, meaning that there is something that we can learn from using specific patterns of metabolites that mimic lifestyle exposure, potentially healthy lifestyle exposure. So in conclusion, I think that these are incredibly complex data, and sometimes if we keep track of what we are doing, I believe that strategies that try to tackle this complexity could be very useful. The potential of omics data is incredible. Sometimes people that deal with very complex model crash against the evidence that the biology in the end is way more complex than any sophisticated statistical modeling. And I think this humble thinking is a very useful attitude for people that try to develop fancy modeling. So what is important, and I think it's something that also Augustin alluded to, is that to create a multidisciplinary setting. This is the only way, in fact, to understand a very complex milieu. This is not a one-man show. If we learn to speak a common language, I think this could be a very decisive element. So I'd like to thank a few people from my group, from Augustin's group, and from Mark Gunter group. Paolo Vinis is always inspirational he has always a lot of ideas. And let me thank you, the audience, and also the organizer. Thank you.
All right, let me start my video. And apologies, where's the share again?
Bottom of the screen.
Thank you much. All right, and can I confirm that this is all showing?
Yeah, it's fine.
All good.
All right, well, thank you for the introduction. When I first accepted the invite, I thought 15 minutes was gonna be enough, but word of warning, it wasn't. This talk is jam-packed. So I'm gonna start with a 5-minute snapshot of the current state of metabolomics and cancer etiology research. And then I'm gonna walk through the results of a breast cancer study of my own. So in the research space of metabolomics and cancer epidemiology, there are now hundreds of studies to date, and there are also hundreds of findings. The research to date has generally focused on colorectal, lung, breast, and liver cancers. There's also been a variety of different sample types examined, and that's what I'm showing in those 2 pie charts to the right of cancer types and sample types. And these are from a review by Leisenfeld et al. But there's a caveat here, which is that virtually all of the studies done to date have been case control. virtually all, meaning probably 85, 90%. And case control studies can suffer from a number of biases. So the first and most obviously is that the cancer itself can affect metabolite levels. That would be, for instance, through cachexia. Another problem is selection bias. That can affect any case control study. Some other problems that are a little bit more specific to metabolomics include that the fasting status or bowel prep might differ by case status. And so we found this to be the case for one of the case control studies that we did. It actually ruined the study. There are differences in the hospital-based diet depending on case status versus control status. And we've actually had a study go belly up on that issue too, found lots of associations. It turned out that many of the controls were at home where they were smoking and drinking and eating their unhealthy diet, causing it to appear that those things were actually protective for cancer. And lastly, I want to mention that sample handling can have a big effect on metabolite levels, and that too can differ by case status. In particular, the nurses that collect samples from cases often differ from the nurses that collect it from controls. And so the process of that handling can vary in subtle but still important ways. And I wanted to end with one additional word of caution here, which is that these effects may not be huge on an absolute basis. So for sample handling, we often find that about 10%, 20% of the metabolites are affected by really rough sample handling by about 10, 15%. But to put that in context, a 14% effect on a metabolite level would be like an odds ratio of Q5 versus Q1 equals 1.9. So what that means is the effect of sample handling can cause associations that are similar in magnitude to those that we actually hope to detect. So that's obviously problematic because it means those associations can obscure the ones you hope to find. So I'm going to focus on the prospective cohort studies because those are the studies that I feel are the most reliable. There are 15 in total to date, covering 6 different cancer sites. I'm going to show you a table of this in just a second, but I wanted to start with some key facts. So first, the median number of cases across these studies was 226. The median number of metabolites was 122 with a maximum of 695. And then for each cancer site, and this is important, there was no overlap between the studies in the terms of the platforms that they used, except for prostate cancer. And each study involved populations of a different nationality. So in terms of replication, in many ways, this is less than ideal. What it points out is that these are still relatively early days. So these are the results of those studies. For colon cancer, the cancer's on the left and the findings are summarized on the right. There were some interesting interesting findings in a Chinese study where they showed that there was decreased risk associated with higher levels of glycerophospholipids, increased risk associated with picolinate levels, but there were no significant associations in studies that were done in the US and Europe. For pancreatic cancer, and I think this is the cancer for which we have the most solid results to date, branched chain amino acids, their levels were associated with increased risk of pancreatic cancer, and this has been validated in at least one study. Plus, I know of one other in process that finds similar results. And then there was one other study in China that again found the same pattern as with colon cancer, lower risk associated with glycerophospholipids, increase with picolinate. Breast, I'm going to talk about in a second in the studies that I'll be presenting. Hepatocellular carcinoma, actually Augustin talked a little bit about already. So there were some associations between tyrosine and phenylalanine increased risk. Strangely enough, branched chain amino acids were associated with decreased risk, and kynurenine was associated with increased risk. For lung cancer, bilirubin was found to be associated with this decreased risk, but because that's just lower in smokers, it's probably related to residual confounding by smoking. And diacetylspermine was associated with increased risk, but only within the 6 months prior to diagnosis. And lastly, for prostate cancer, there have been suggestive associations, but none were significant after multiple testing. There's a little bit of a hint that alpha-ketoglutarate levels might have a consistent association across populations, but that's still relatively, crude results, early results. So with all that said, what we're left with, I think, is a relatively small set of findings to date. and I've summarized those here. So the branched chain amino acid association, I would say, is the only validated or replicated association. But I want to caution that these are early days and we had very, very low sample sizes. So when you account for multiple testing and other factors, you need large sample sizes to detect associations in these studies. I'd also add that there's some very promising leads, and more importantly, perhaps, the studies are growing fast. So I have seen in my own experience that studies with tens of thousands of participants are becoming the norm. That includes even for some grants I've seen go through for Metabolon, which Augustin showed that the sample sizes to date aren't that large. The capabilities of metabolomics platforms are also improving quickly, and that's attracting the interest of more PIs. So if there's maybe 100,000 participants in prospective cohorts today that have metabolomics data, we can expect that number to keep increasing and increase rapidly. And in terms of the capabilities, I just wanted to highlight this one idea briefly. On the right, I'm showing a figure of the number of identified metabolites, that's on the y-axis, by study year, that's the x-axis, for 23 different studies that my division has done with the same metabolomics provider. And you can see that when we started our research in this area back in right around the beginning of 2012, we were only getting about 400 metabolites a study, and the last one that we've done gives us about 1,200 metabolites. So this is doubling every 4 to 5 years, I estimate. All right, so that was very high level and abstract. I'm going to give a more specific sense of metabolomics in practice by showing results from a study that I conducted, and this was published as 2 different papers. My fellow and I actually divided this based on metabolites that were related to body mass index and metabolites that were related to diet. And we did this in part because we had seen from prior studies that those 2 sets of metabolites actually don't have that much overlap and represented logical discrete units to split this up on. All right, so first the BMI study. This study was done in the PLCO, which is a large prospective cohort. I included 621 invasive cases and 621 matched controls. 418 of these were ER positive. The median time from sample to diagnosis was about 7 years, and the controls were matched on standard factors. The statistical analysis here was done in 2 stages. So we analyzed the correlations between body mass index and metabolites first, adjusted for certain factors, and then we analyzed the association for that set of BMI-related metabolites and their relationship with breast cancer risk, again, adjusted for standard factors. And on the right, I'm showing the flow of the metabolomics assay. So starting with year 1 serum, it's non-fasting, going to the LC mass spec and metabolon, and ending with 617 identified metabolites. So this is the first part of that analysis. It's a Manhattan plot of the BMI metabolite associations. On the x-axis, we have the 617 metabolites separated by chemical class. On the y-axis, the p-value. And there is a gray line going across, which is our threshold of statistical significance, which is the Bonferroni threshold. But we also additionally required that metabolites had to have a correlation value of 0.15 or higher, or negative 0.15 or lower. So in total, 67 metabolites met that threshold. Just to give you a quick sense of what some of these things are. So the first example here is valine. This is one of the 3 branched chain amino acids. So one of the ones from the pancreatic cancer study. It's also been associated with increased breast cancer risk recently in the SUVIMAX cohort, so definitely relevant here. The second is mannose. It's a glucose epimer that appears to outperform fasting glucose for predicting future diabetes risk. The third is gamma-tocopherol, and it's basically a proxy for poor diet. Its levels appear to reflect consumption of corn and other vegetable oils like you might have in fried foods. And lastly, hydrochlorothiazide is a drug for high blood pressure. So I'm showing this just to give a sense of the kinds of things that you can pick up from this assay and to give a sense of the metabolites I'm examining. So overall, 6 of the BMI-related metabolites were associated with risk of breast cancer, and that's what this plot shows. The false discovery rate was less than 0.2 for each of these. Odds ratios comparing a high level, that would be at the 90th percentile, versus a low level, approximately at the 10th. These odds ratios were about 1.5 to 1.8, which is similar to what you get in other biomarkers of breast cancer, for instance. So estradiol, I think, will have a Q5, Q1 odds ratio of about 2. So kind of in that ballpark range. These metabolites were all higher in women with higher BMIs, so the data are lining up in the expected direction. We next found 23 metabolites associated with the risk of ER-positive breast cancer. Odds ratios were a little higher than for overall breast cancer. And again, the directions of effect were consistent with the direction of the BMI association. But perhaps more importantly, we have here the metabolites that are independently associated with breast cancer risk and also ER breast cancer risk. So we know that metabolites are often highly intercorrelated. One way of addressing that very simply in a model is just to put everything in the model and identify what comes out at the end. I can go into a little bit more detail about how we did that, but I'm going to focus on the results for now. What we had at the end of this process were 2 metabolites with mutually independent associations with breast cancer. So that's what this is showing. And we had 4 metabolites that were associated with ER-positive breast cancer. So that's what this is showing here. And I'm going to go into each of these briefly. So our top hit was this metabolite 16-alpha-hydroxy-DHEA-3 sulfate that's highlighted in bold. It's part of the sex steroid hormone pathway, which is what I'm showing in the figure to the right. My initial reaction to this was not excitement. I was thinking, oh Lord, we discovered something that people have already discovered. But as I looked into this more, I began to appreciate this result in a much greater way. So this 16-alpha-hydroxy-DHEA-3 sulfate, it's essentially a metabolite that's produced on a process that's parallel to the process that produces estradiol. So estradiol, for instance, is down here on this other corner of the box. We also just examined this more directly. Instead of thinking biologically, we just said, hey, what's the correlation? And 16-alpha-DHEA-3 sulfate had no correlation with estradiol. In our data set, we had estradiol measured in 260 of these. The Pearson correlation was negative 0.04. So that was striking because we had actually never seen that with a sex steroid hormone before. Usually they're all tightly intercorrelated. We also examined this in an independent replication data set from Canada and had a R of 0.00. So these really are uncorrelated and represent distinct phenomena. The other 3 metabolites, interestingly, they all clustered into one metabolic pathway, which I'm showing here, and that is the degradation of isoleucine and leucine. So walking through the 3 metabolites that we picked up, alloisoleucine, for instance, accumulates in response to low activity of the branched chain 2 oxoacid dehydrogenase enzyme. Each of these amino acids that I'm about to show accumulate in response to low activity of some enzyme. So 2-methylbutyryl carnitine accumulates in response to low activity of this 2-methylbutyryl CoA enzyme, and 3-methylglutarol carnitine accumulates in response to low activity of this 3-hydroxy 3-methylglutarol enzyme. And so this is interesting because it does tend to support this idea that branched chain amino acid metabolism is important for cancer risk, maybe for breast cancer as well as pancreatic cancer. And so moving to the diet part, I'm showing you now that this is the same design as what I showed you before with one exception, and that's in the box here. It's just the statistical analysis part that varies. So same cases, same controls, same matching, same metabolomics assay. But what does differ is that we first begin with analyzing the correlations of 55 foods and beverages with metabolites adjusted for standard factors. Then we analyze the associations between these diet-related metabolites and breast cancer risk adjusted for standard factors. So overall, 34 of the 55 foods that we examined were associated with at least one metabolite, and 113 metabolites were associated with at least one food. And putting it all together, you actually get 222 total correlations, because some foods were associated with more than one metabolite, and some metabolites were associated with more than one food. So 222 total correlations. I'm not going to go through all of them, but I will say this: the most we observed was for coffee intake, 32 metabolites. Many of these observations replicate what we had observed before, except that the magnitude of the correlations was actually a little bit stronger than what we saw before, possibly reflecting some improvement in the assay. So our top metabolites were getting correlations benchmarked against an FFQ of 0.6, which seems quite reasonable on its face. Now this is a bit of a spoiler, but none of these were associated with breast cancer. So that's the last you'll hear of them today. There were also quite a few metabolites associated with drinking alcohol, and of these, quite a few were related to androgen metabolism. In terms of our thinking on this, it's not that we think that alcohol has androgens in it, and therefore when you drink it, your levels increase. It seems to be more of an effect on your endogenous metabolism. It causes an increase in the production of androgen-related metabolites. So I just wanted to make sure that that distinction was clear between an exposure biomarker and a biomarker that is the result of the food's biological effect. So the top 20 associations for overall breast cancer are here. Only the top 3 are statistically significant at the FDR 0.2 level. So these were metabolites that were associated with butter. So the top metabolite is caprate. It's a medium chain saturated fatty acid found in dairy fat. Next was a metabolite associated with vegetable oil or vitamin E, and that's a gamma CEHC. And this is a excretion product essentially of gamma tocopherol itself. Gamma tocopherol is found in vegetable oils, as I mentioned before, in the BMI study part. And third, there is a alcohol-associated metabolite, this 4-androsten 3-beta, and it's androgen related. The top 20 for ER-positive breast cancer are shown here. Odds ratios here are ranging up to 2, so in a sense similar to the BMI study, we saw stronger effects for ER-positive breast cancer. 12 alcohol-associated metabolites. Most, but not all of these, are related to sex steroid hormone metabolism. Again, we had caprate and 2 other metabolites associated with butter and fried foods. And we had tocopherols, including gamma tocopherol, or the gamma CEHC rather, and alpha tocopherol. So in short, 3 dietary fat-related metabolites were associated with breast cancer. They're listed here. The reason we might be interested in this is that there is a growing body of evidence suggesting the fatty acids may be heterogeneous with respect to their association with breast cancer. And one of the things that I find interesting about metabolomics is that it allows you to assess these kinds of fats at a level of granularity that we haven't really had before. It's not possible in the FFQ because many of these things aren't in a nutrient database, for instance. So it allows us to explore exposures that we hadn't previously explored. The tocopherol metabolites that were associated with breast cancer are shown here. Tocopherols have a sort of conflicting literature with respect to cancer risk. Our results are not going to resolve those conflicts, but the results that we did see were consistent with what we would expect based on how each of these metabolites tracks with either a healthy or unhealthy diet. So they may be serving as a proxy for the kind of healthiness of diet. And lastly, the alcohol-associated metabolites. These are primarily sulfates of DHEA or androstenedione. There is strong evidence indicating that consumption of alcohol increases levels of DHEA and metabolites that come after that, based on a feeding study. The contribution of our study specifically is it suggests other downstream metabolites that could either be explored or analyzed in relation to breast cancer. And we did find some mechanisms that appear to be hormone independent. So for instance, there was a metabolite alpha hydroxyisovalerate that had no relationship to any of the sex steroid hormones, but was related to alcohol and breast cancer risk. So maybe there's another pathway there that could be explored. So with that, I come to this summary here. I'm not going to read it except to say that, as always, future or further replication studies are needed. These are early days, and I think it's really important to conduct bigger and better studies to keep pushing this area forward. So I'd like to acknowledge my collaborators, and thank you very much.
Hello, well, this is Marji speaking, and I just wanted to thank all of the speakers for their excellent and very informative presentations today, and for the audience members to be here. I don't see any questions from the audience, so I'm going to ask one of my own, and actually I have about 10, but I will ask one, and that is, there are many, it seems to be many promising avenues for the area of metabolomics and both diet biomarker and cancer association studies. And for the former, for diet biomarkers, if you discover interesting biomarkers, as was shown by a couple of the presenters, that seem promising for, say, coffee or alcohol, what do you think we need to do to be able to confirm that they are good biomarkers to use in place of alcohol, say? I know that there are relative concentrations for untargeted metabolomics. So I just wanted to see if one or more of the speakers could speak to that.
Probably all of us, I think, have opinions on this. I will start, if that's okay, because I think I can keep it pretty short, which is to say that I think that we ultimately need to be doing some kind of feeding studies where we're essentially weighing food to the gram, and then examining the magnitude of the correlation, with these biomarkers. So if you have a gold standard measurement and you obtain correlations that are very high, and if certain other properties, important properties of biomarkers are met, then you can assume that it's a good biomarker. I think the really hard part in this is determining the time scale over which a biomarker operates. And so there may need to be some maybe rethinking of feeding studies and how we conceptualize that issue of time scale. A lot of our, what we call gold standard biomarkers are recovery biomarkers, meaning that you can literally recover them over a 2-week observation period. But those metabolites may not necessarily be optimal for longer term assessment of diet.
Thanks. Did anyone else want to comment on that one?
I just want to remind the speakers before, I'm sorry to interrupt, that they can type questions. I don't know if people joined later and they didn't know, so you can actually go to the Q&A button at the bottom of the screen and type your questions, and we will be able to read it. Sorry, you can go ahead. Augustin?
Yeah.
Okay.
Maybe about dietary biomarker, I think that Steve has mentioned some key aspects for validation of biomarkers. One difficult question is also about dietary biomarkers is their specificity can they be confounded by other dietary source, and it is a question that is not easily answered because it requires a global view actually on all possible confounders that we may have in the diet. So we learn a lot about the structure of the biomarker and the current knowledge we have on the chemistry of foods, and this information is sometimes found in some database and sometimes cannot be found in a database, and so this remains sometimes an issue to conclude.
Thank you. And Pietro, I was struck by the difference in the association between the metabolomics signature and the healthy lifestyle score, and I was just wondering if you have come up with some ideas of interpretation for those findings.
You're right. We did our best to try to kill that association because in fact, we couldn't really believe why it was so much stronger than what we observe with standard assessment methods. Honestly, I don't have an explanation. It's true that it's probably the fact that we were capturing something that was the results of 150 variables, as compared to lifestyle indicators. Even if these are incredibly powerful, but it's probably there is something in the data that we didn't manage to understand yet. It's true that now that I think of, we should probably run some sensitivity analysis, try to understand what are the drivers of such inverse associations, and yeah, this is something clearly to understand.
Okay, well, thank you. And I want to point out that it is 4:05, and this is scheduled to end at 4, but I'm going to go just a couple more minutes to answer a question that, to ask a question that was just posed of the speakers, and it is it meaningful to analyze metabolomic signature in older samples from existing studies without diet questionnaires? One of the speakers.
Yeah, clearly, for sure. I mean, it's clearly if you have it. I mean, I guess the question Sam is asking is actually it's probably not Sam, but anyway, I mean, you clearly need to have an outcome. So it will be like a cancer indicator. That will be idea. You do not necessarily need to have questionnaires. Clearly, if you have them, then it means that you increase the possibility of characterizing what you are observing. But I think you could do an annotation work independent, irrespectively of the fact that if you have questionnaires or not.
Okay, thank you. Well, I think, let's see, there
is one question.
Sam did ask a question, and that is, any idea of the best technology for untargeted metabolomics and cost?
Yeah, I think that we can discuss this at length, because there are so many options today still, but I think that the technology that offers the wider coverage in terms of number of metabolites that can be detected and then eventually identified is clearly high-resolution mass spectrometry coupled to liquid chromatography. This doesn't mean this is the only technology. Gas chromatography coupled to mass spectrometry is also another technology. But this the liquid chromatography coupled to high-resolution mass spectrometry is the one with the highest coverage. And then about the cost, because the question was raised as well, and the costs vary obviously according to the companies and the technology. If you collaborate with an academic lab, then the cost would obviously be also different. Just to give you an order of magnitude of the cost, it may vary between 100 to 400 US dollars. I don't know, Steve, you may have also some other information on this side.
Yeah, 200 to 350 is fairly typical for you know, a metabolomics lab that's providing mostly identified metabolites in at least 350
per sample.
Per sample. Mm-hmm. Yeah, somewhere in that ballpark.
Great. Okay, well, thank you again. I think we can close here. I just want to thank all of the speakers for their excellent presentations and their insights into this complex and growing field. And thanks to the audience members for participating. And I guess with that, we'll just sign off.
I just want to say one thing. Again, thank you to everybody, but also that the recording will be posted in the ASPO website. So anybody that wants to go back and go through it, or you can forward the link to anybody that is interested. Thank you so much, everybody.
All right, thank you.
Thank you very much.
Thank you. It was a pleasure.